Lizard Bio Wizard.
Guys, I would like to introduce you to Lizard Bio Wizard.
Did you know that all living things are composed of organic molecules? Organic molecules can be separated into 4 main categories: lipids, carbohydrates, proteins, and nucleic acids. Let's start with carbohydrates. Carbohydrates are made up of monosaccharides, which are simple sugars. Examples of monosaccharides are glucose and fructose. The difference between mono, di, and poly is simply how many sugars there are. Disaccharides and polysaccharides are created through the processes of dehydration synthesis. Dehydration synthesis occurs when a water molecule is removed from 2 monosaccharides, resulting in the formation of a covalent bond, which is called a 1:4 glycosidic linkage. The opposite reaction is hydrolysis. Hydrolysis is when you add water between the covalent bond to break up the polymers. This happens during digestion. Here, you see my assistant Julia and me golfing. In order to do this, she needs energy. There are 2 different types of carbs, storage and structural. Storage carbs store energy, and structural carbs provide support to their cell. Switching molecules, let's talk about lipids. Lipids are long hydrocarbon chains that are hydrophobic, and they include glycerol and fatty acid tail. Their functions include providing structure and storing long-term energy. Hey Julia, do you know the different types of lipids?
I think it's fats, oils, phospholipids, and steroids.
Right, but don't forget waxes. The difference between the types of lipids are that waxes contain alcohol. Steroids are 4 fused carbon rings, and phospholipids contain a phosphate group and a second fatty acid tail. Saturated fats are single bonded and solid at room temperature. Unsaturated fats are liquid at room temperature. Let's talk about proteins. Proteins monomer are amino acids in which the R group is what makes an amino acid different from others. Julia, how many different amino acids are there?
20.
Yes! A protein has 4 different structures, and the primary contains different amino acids that make up a protein. Only peptide bonds are present in this structure. Next, we have the secondary structure, which includes the first folding of proteins where it will become either an alpha helix if the protein needs flexibility, or a beta sheet if the protein needs to be strong. Only a peptide and hydrogen bonds are present in this structure, which allows the protein to bend. And the tertiary structure, folds, which determine the shape of the protein, are present due to the interactions between the R groups. Quaternary structure differs from tertiary because more than one polypeptide sequence is folded together.
Liz, aren't we forgetting one type of molecule?
Why, yes, my friend, nucleic acids. You are probably familiar with DNA and its double helix. Okay, well, we know that it contains our genetic makeup, and it's an example of a nucleic acid. By looking at this molecule, you see that there is a phosphate group, a sugar, and nitrogenous bases in each nucleotide. A nucleotide is the monomer of nucleic acid. It is important to note that not every nitrogenous base is the same. There are 5 different bases. Adenine, guanine are purines, and cytosine, thymine, and uracil are pyrimidines. The different sequencing of these 4 base pairs accounts for the diversity of life. It is what makes a human different from a chimp. A purine must always pair with a pyrimidine. Guanine pairs with cytosine, and adenine pairs with thymine or uracil if we're looking at RNA. Another important nucleic acid is ATP, which provides energy for animal and plant cells.
Thanks, Lizard Bio Wizard. Alright kids, that's all you need to know about organic molecules. Good luck on your AP exam.